talking about togetherness.
Hello, welcome everybody to this third day of the research discussions on HAT and where are the actual gaps in our knowledge which could improve movement towards very, very low prevalence over the coming decade and try to achieve the WHO objectives of elimination by 2030. I'm going to be very brief in this introduction just to remind everybody the real objective of today's session is to end up with a very short list, as it were, of gaps in our knowledge where we think research could really accelerate progress towards elimination, or at least reducing the burden of disease to very, very low levels. Now, it's my real pleasure to hand over now to Florent, who is going to chair these discussions, and then we will all get together at the end to try and rank these research priorities. Florent, over to you.
Hi, everyone. Thank you very much, Roy. Welcome to all participants to this last session of the scientific meetings. I'm going to present the panelists of this session, started by me. My name is Florent. I am the HAT platform coordinator, and I am also in charge of access. I'm working in DNDi-HAT. I want to introduce the second panelist, Professor Simon Croft. He has worked on discovery and development of anti-effective drugs, and also the research of Simon focuses on novel drugs and formulation for the treatment of leishmania, malaria, human African trypanosomiasis, and Chagas. José Ramón Franco from WHO is working in Department of Control of NTD in WHO Geneva. It is a medical doctor. In this position, he gives a technical and financial support to different endemic countries, coordination of countries and partners, development of technical reports and guidelines. He is ensuring access to HAT treatment and pharmacovigilance of new developed HAT medicine, development of clinical trials to new medicine and new diagnostic tools, in the setup of WHO Atlas of HAT training of health staff, including the international course for African trypanosomiasis. The last is Paulo Macana from Institute for Control and Combat of Trypanosomiasis in Angola. He is a medical doctor. He is a head of Department of Technical Management and Supervision of Institute of Combating and Control of Trypanosomiasis in Angola. He has experience on the impact of mini mobile team in Angola and the integration of diagnostic and the treatment activity into the national health system. I'm going to share the slide so that you can see the The objective of this session is to summarize the point discussed in the two previous roundtable, and also with the priority reported with the research gap, which can be investigated and deepened for HAT elimination by 2030. During the first roundtable, for the two previous roundtable, I'm going to talk in French, but the slides are in English. Je vais parler en français pour nos amis francophones. Et comme les slides sont en anglais, ça va permettre à nos amis anglophones de bien suivre. Les éléments qui ont été évoqués et les sujets qui ont été discutés lors de cette table ronde, c'était au sujet des outils de surveillance et des outils de diagnostic. On aimerait que tous ces outils soient des outils simples et adaptés au terrain avec une bonne spécificité et une bonne sensibilité. Il y a aussi besoin d'adapter la recherche active des cas au niveau des pays endémiques et voir dans quelle mesure on peut rationaliser le dépistage actif ensemble avec le dépistage passif. Un autre point qui a été abordé, c'est au sujet de tests moléculaires pour la trypanosomiase. Et là, les panélistes de cette session ont évoqué qu'il y a une faible information sur la performance de ces tests en termes de sensibilité et de spécificité. Et il y a une difficulté de mettre en œuvre ces tests dans des milieux reculés. Et surtout, comme vous le savez, dans ces milieux reculés, les infrastructures n'ont pas assez d'équipements pour leur permettre de réaliser ces tests moléculaires. D'où il faut que des études soient menées dans ces milieux pour permettre de valider d'éventuels candidats pour ces examens. Et il y a aussi besoin de coupler les essais cliniques pour assurer un bon suivi sur le terrain. L'importance du réservoir animal avait été aussi mentionnée et les experts ont parlé plus sur les diagnostics des animaux domestiques par l'utilisation des tests moléculaires, mais l'on constate que ces tests sont souvent pas tellement efficaces et coûtent cher. Et d'autres ont évoqué l'utilisation des insecticides auprès des animaux afin de couper la chaîne de transmission entre les mouches et les animaux domestiques. Et l'importance de la migration de vecteurs de mouches tchétchés a été également mentionnée. Et on a estimé qu'il fallait comprendre plus les mouvements de ces mouches et voir comment anticiper sur la réinvasion des espaces déjà nettoyés et éviter qu'on puisse assister à une augmentation de la densité glossinienne dans les zones où le contrôle vectoriel pourra être arrêté.
Excuse-moi, Florent, on ne voit pas les slides. Hein?
Les slides?
On ne le voit pas.
OK. Que je les partage. OK maintenant?
Non, pas
Ça
encore.
va? Pas encore.
Maintenant, merci.
OK. OK. Okay, I was saying about the first roundtable. Je disais que lors de la première table ronde qui était consacrée au diagnostic, à la surveillance et au traitement et au contrôle vectoriel, il a été mentionné lors des discussions que on doit beaucoup travailler sur les outils de diagnostic et de surveillance qui doivent être simples, bien adaptés sur les terrains et avec une bonne spécificité et sensibilité. Et il y avait aussi besoin d'adapter les activités de dépistage et comment rationaliser le dépistage passif avec le dépistage actif. Et on a évoqué également le problème des tests moléculaires pour la trypanosomiase et les experts ont relevé une faible information sur la performance de ces tests, surtout les tests PCR, en termes de sensibilité et de spécificité. Et il y avait la difficulté de mettre en œuvre ces méthodes de diagnostic dans les milieux reculés et dans nos pays endémiques, surtout avec une infrastructure pauvre. Et il a été proposé que de recherches puissent être menées dans ces milieux pour valider ces candidats tests. Et les essais cliniques également devraient être couplés avec la réalisation de ces méthodes de diagnostic sur le terrain pour assurer un bon suivi des malades au niveau de nos villages. Et l'importance du réservoir animal a été mentionnée également dans la transmission. On a beaucoup plus parlé des diagnostics des animaux domestiques avec des tests moléculaires, mais certains experts ont évoqué que l'efficacité de ces tests de dépistage et ces tests coûtaient un peu plus cher. D'autres ont évoqué le traitement de l'imprégnation des animaux domestiques avec des insecticides pour couper la transmission entre les mouches et le sang des animaux domestiques. Le problème sur la migration des vecteurs a été également mentionné et les experts ont dit qu'il fallait comprendre plus le mouvement des mouches et voir comment on peut anticiper la réinvasion des espaces déjà nettoyés et éviter un peu l'augmentation de la densité glossinienne dans les zones où le contrôle vectoriel pourra être arrêté. Et le contrôle vectoriel pourra être considérée comme une intervention pour protéger également les populations vulnérables et surtout les réfugiés et les déplacés des guerres dans des zones endémiques. Les interventions également de contrôle vectoriel peuvent être mises en œuvre pour soutenir également les interventions médicales qui sont réalisées par les programmes de santé. On a mentionné également l'effet de changement climatique sur la transmission en disant que dans certains coins, les hautes températures avaient une influence sur la densité glossinienne et cela aussi pourrait entraîner également une augmentation de la densité glossinienne dans certains milieux. Et le rôle de COVID-19 a été mentionné et on a évoqué que cette pandémie a essayé également d'impacter sur l'élimination de la trypanosomiase avec l'interruption des activités de dépistage dans les pays endémiques. Le deuxième jour, c'était la table ronde sur les sciences sociales. Et durant cette table ronde, on a évoqué l'importance de l'engagement communautaire. Et pour cet engagement communautaire, on a relevé que les connaissances et les attitudes de la maladie commencent à changer avec la baisse de la prévalence, puisque les mêmes connaissances qu'on avait lors des prévalences élevées ne sont pas les mêmes auprès des populations avec la rareté de la maladie. Et c'est pour cela on a dit que des études peuvent être menées pour voir comment les communautés peuvent être engagées. Par exemple, les leaders communautaires, les églises, les leaders traditionnels dans ce travail de communication sur la THA. Et aussi, les experts, les panélistes lors de cette table ronde ont évoqué également les connaissances des populations locales au niveau du programme sont souvent, ne sont pas reconnues et souvent nos programmes nationaux imposent leurs connaissances aux communautés et il devrait y avoir un dialogue réciproque entre les programmes et les communautés afin qu'il y ait un échange pour que les programmes aussi à leur tour puissent avoir un œil sur ce que disent les communautés en rapport avec la maladie du sommeil. Et c'est pour cela les études doivent être menées pour voir comment améliorer cette collaboration et comment reconnaître que ces connaissances locales est aussi scientifique et réelle et logique. Et les communications, les messages pour la communication doivent être réalisés également au niveau des communautés par des personnes du milieu qui sont connues et qui sont respectées pour faciliter l'assimilation de messages liés à la trypanosomiase par nos populations respectives. Et concernant les ressources humaines, on a relevé également la confiance qu'a la population ou les populations touchées aux personnels de santé qui sont impliqués dans le traitement de la trypanosomiase par rapport aux autres personnels de santé qui ont moins de capacités par rapport à la THA. Et aussi, la confiance de la communauté est très importante pour les médicaments et pour les tests diagnostiques que nous utilisons. Et souvent, les populations ont plus confiance aux médicaments oraux qu'injectables. Et dans ces cas, déjà, les avancées significatives ont été obtenues avec l'avènement de nouveaux traitements comme le fexinidazole, qui est un traitement oral et qui puisse améliorer la confiance également de la population par rapport aux outils diagnostiques tant thérapeutiques qui sont utilisés. Et également, il a été proposé qu'il faut qu'il y ait un renforcement des capacités des autres prestataires et pour essayer également à les amener à s'impliquer dans la prise en charge de la trypanosomiase dans nos communautés. Et aussi, des études pourront être menées pour comprendre comment nos communautés, nos populations voient nos prestataires par rapport à leur façon de délivrer les soins au niveau des pays endémiques. Aussi, les experts ont relevé également que les données chiffrées ne doivent pas occulter l'expérience de nos populations affectées et qui souvent restent avec leur stigmatisation et parfois qui n'est pas prise en charge par les programmes en dehors du traitement qui est donné. Et rarement, il y a le suivi de tous ces problèmes sociaux que rencontrent nos patients sur le terrain. Et comme la prévalence est en train de diminuer et la perception également du risque est en train de disparaître, il y a besoin de combiner les activités de la trypanosomiase avec les autres activités pour essayer d'attirer la population à s'impliquer dans les activités de lutte contre la trypanosomiase. Et également aussi une approche multidisciplinaire doivent être considérées impliquant et le monde humain, l'environnement et également le monde animal pour que cet aspect de lutte contre la trypanosomiase soit pris de façon multidisciplinaire. Certaines gaps en matière de recherche ont été identifiés. Par exemple, bien faire la cartographie des acteurs au niveau communautaire qui peuvent s'impliquer et identifier les moyens de donner, de délivrer la sensibilisation en impliquant les élèves dans les écoles et aussi d'autres membres de la communauté. Et voir comment améliorer les effets psychosociaux sur la trypanosomiase et lutter contre les effets de la stigmatisation sur de la THA au niveau des populations touchées. Et aussi, les experts ont parlé un peu des technologies qu'on pourrait mettre à la disposition des communautés pour avoir accès à l'information sanitaire. Et cela pourra permettre à nos populations d'être en contact avec l'information sanitaire et aussi de s'impliquer dans cet engagement communautaire. Donc, voilà résumé les deux tables rondes que nous avons eues ensemble. Now we are going to ask the question to our panelists. What can our panelists say about the priorities and prospect for HAT elimination by 2030? I'm going to introduce Dr. José Ramón. WHO worked a lot about the priorities and the prospect for HAT elimination 10 years ago. They give us the guideline. They give the National Sleeping Control Program the guideline about the priorities and the prospects for HAT elimination. Maybe Dr. José Ramón can add something about this. José Ramón.
Thank you very much, Florent. Very kind. Well, I think it's just a very general question, no, to start the discussion today. What can I say? Well, the main thing is that we started to reinforce this control program 20 years ago when the situation was completely different. Thanks to the effort of all of us, and mainly national control programs, but all the other people and new tools that they have been coming, we have advanced a lot in the control of the disease. And in a certain moment, we have reached a situation that we consider that sleeping sickness is not anymore a public health problem. It's not only a title, it means that it's not feel like it's an important problem anymore. But we can't stop here. We have to keep on moving, we have to go further, and for that, we need to change, we need to adapt things. Sleeping sickness has been a disease that, well, all the control has been done with tools that they were not perfect. We can't aspire to get perfect tools. But if we have more adaptive tools, we can say better tools, it will facilitate to reach this new goal we have established, that is the elimination. It means to reach that there is no transmission of the disease, that there are 0 cases of the disease. For that, new tools can help a lot. But we have to change also the way of doing things. We are changing in a progressive way the way of doing things. Before, we were screening a lot of people, diagnosing a lot of cases, treating a lot of cases. And now this has become, we screen a lot of people, but we find few cases. We don't need to treat big amounts of people. And this is becoming a challenge. So I think all this research has to be addressing this challenge we have today. It has been talked during these days a lot about we are losing all the expertise. We had before people working in the field with a lot of knowledge about the disease, but because, well, the time where it has passed, many of them, they are going to retirement, and we are losing this expertise. And it's not possible to recover this expertise because we don't have so many cases. So we need to adapt to the tools in the future to people with less expertise. It's for that we are talking about mainly in diagnosis, about tools that they are easier to use at the field level, but we can keep this expertise in some referral centers. Then it's important, I think it was talked the first day, how we are sampling these samples at the field level to be sent to the higher level, to the referral levels, that they can do more sophisticated tests that, as you have mentioned, it's not always possible to put at the field level. That's one thing I think is important. The same in the treatment. We are doing important steps to go for treatments that they are easier to use, that we don't need specialized people knowing these sophisticated drugs as melarsoprol or as nifurtimox or as eflornithine. We need now drugs that they can be used easier. So we are moving in this sense. It has been talked also that now we are getting easier tools to use, performance tools for diagnosis, but they are not sensitive enough. But if we have drugs that they are less toxic and they are effective, we can approach this issue to treat people that they are not confirmed as cases. But this, we need to invest in that, to do trials to see that these drugs are safe enough. It's a general idea just to start. I think my colleagues also, they have many other things to say. Thank you.
Thank you, Dr. José Ramón. I go to Professor Simon. Do you have something to add?
Thank you, Florent. Yes, I agree with what Jose has just been saying. I think it was on the first day, there was a lot of discussion on the difficulties of surveillance and the importance of surveillance, and so the difficulties of when you've got diagnostics which are either not sensitive or specific enough. But what really wasn't discussed much on the first day was actually the new treatments coming along. And there's only cursory mention by the chairman of the new treatments. And Jose has just referred to this potential of a simple treatment that can be used at community level. And I think through DNDi, the organization that you work for, Florent, and I worked for at one time, we are on the brink of something quite revolutionary with this acoziborol, which you know about and José will know about, the potential of a single treatment for first and second stage disease. The drug acoziborol has both completed phase 3 clinical trials, and so we wait to hear, and an 18-month follow-up, we wait to hear all the details. But the important thing here is, on one side, there is a drug which can be used at the community level. And as José was really pointing out, the issue then becomes, and not one of the effectiveness of treatment, it becomes one of the safety issues. Can this drug be used in people who are within communities who are not actually visually infected or actually show, positive diagnostic? Can it be used at a community level as a sort of a chemoprophylactic? And I think that is a big question. It comes with another important caveat here, which is that the discussions between DNDi and Sanofi, and Sanofi seem to be making a commitment to this drug. And that's very important because it's not just the research that goes on around the use of the drug and its potential at the community level, say in treatment as well as prevention, but also to ensure that there is something which I think does require research in this area, research around a sustainability agenda, because the point is not that we just get the elimination of public health, as already José said, not a public health problem anymore, HAT. Well, to keep it as not a public health problem anymore, there has to be issues around sustainability addressed, and a research agenda around sustainability has to be put into place. So I will leave it at that for the moment, Florent.
Thank you, Professor Simon. I return to José Ramón. Maybe you can give us some gaps in terms of vector control, how we can use this strategy combining with active and passive screening. Yeah.
Thank you, Florent. Well, to say that, yeah, the control of the disease, it has been based during all these years in three pillars. One has been the case detection, another has been the case management, it means the treatment of the cases, and a third pillar is the vector control. We have tools of vector control, but we have to understand that vector control is not to end the flies. It has to be, now we have easier tools to use, that's important. But the important thing now is to know how to use these tools, sorry. We have these tools and we need to adapt to the different circumstances. The transmission is not the same everywhere. We have areas that the transmission happens in mangrove areas. We have other areas that is maybe in cocoa plantations. In other areas is in riverine forest. And the tools we have to be adapted to these different situations. At the same time, we can't cover huge extensions because it will become too expensive and difficult to use. So it's very important that we have better tools to target well where we are doing the vector control, where vector control can get an important impact. So that's important. So also, well, you have seen that there are different approaches. It has been interesting. Maybe it was not so much talk during the social science panel, but there are some interesting efforts now to see how community can appropriate or can handle the vector control. Maybe this is an interesting approach, and I think there is important research going in this way. I think we have tools. There are always room for improvement, but mainly is to address how to use these tools, vector control tools we have now. Also, well, we can't just, we have this interesting tool we have been using a lot in the last years, is the tiny targets, but we can't forget other tools that can be interesting in some settings. For instance, these protective impregnated fences that can be used in areas where there are domestic animals like pigs, that they can play a role in the transmission, or other tools that we need they can be used in different settings.
Thank you, Dr. José Ramón. Professor Simon, do you have anything to add?
I haven't worked on tsetse flies for about 30 years, so I will pass on the vector control side. But I'd like to come back to this more general question, which really comes from day 2. And there was a lot of discussion about how what research could be involved in ensuring that there is community participation. I think these were very important points. And there is obviously other programs for other NTDs which have employed the sort of the behavioral change strategies to try and ensure people are aware of the disease and what to do about it. But what is often missed out, and I have to ask, include José in this, because his wonderful review in PLOS NTD, one of the maps shows how the distribution of diagnostic and treatment centers in the endemic regions. I think this is a very important map, because the question isn't about just the distribution of the maps, but it's how these centers interact with health systems. And I think the WHO has to work with the Ministry of Health and the health systems in the countries, and the health systems actually organize the centers. And what has come up already from José is the losing of the expertise. And it's not just losing expertise like, of the gray-haired older scientists that we have around, but it's also, as mentioned yesterday, the turnover of staff. It's not just losing them because they're getting older, it's losing them because they're being attracted to other positions. So how the health systems work and how to maintain trained staff who can recognize the disease when it occurs, however rarely in the future, and to ensure that there are the things there. And one of the questions that came up on the chat line was about platforms, and it's how these can be used to ensure that there are trained staff within those centers, and those platforms are kept up to date, so the staff, if they are turned over rapidly, can actually access the updated information. And I think how the communities, the health systems, and then the patients interact together is something that really has to be understood more clearly in regions where access is very difficult, and ensuring there is this maintained education of trained staff. I think it's something that José has been involved with, and I think it's an important question to answer.
Okay, now I will give the word to Dr. Macana. He will speak in French. Dr. Macana, je voudrais vous poser la question. Vous qui travaillez dans le programme de contrôle sur le terrain, on aimerait savoir comment vous essayez de rationaliser le dépistage actif combiné avec les activités du dépistage passif et également avec le contrôle de vecteurs sur le terrain. est-ce que vous avez aussi quelques déficits en matière, quelques difficultés qui pourraient faire l'objet de recherches sur le terrain par rapport à l'implémentation de vos activités de lutte sur le terrain? Voilà. Docteur Macana.
We're not hearing this.
Docteur Macana, le son ne sort pas très bien. Je ne sais pas, est-ce que vous avez un problème de micro? Essaye d'arranger le micro. On va revenir vers vous pour que vous puissiez revenir. Arranger un peu le son on va revenir vers vous dans 2 ou 3 minutes. Donc, nous allons aller dans des questions. We have 2 questions for José, Ramón, and Professor Simon. Do you envisage a scenario in which a single dose available treatment, easy to use and safe, could be used massively, such as PCNTDs in the communities where transmissions persist despite effort to interrupt it? Do you think that this could be feasible if a suitable treatment, acosiborol, would be available? You can see the question and you can answer. Maybe Dr. José Ramón can start.
Thank you, Florent. Thank you for the question. We have to see that we have some differences with other diseases that they are using this mass drug administration. We are talking of, and currently now we are talking about very low prevalences of the disease. So there are few real cases of the disease. So if we are using in an extended use, it has to be a very safe drug, because we'll be treating many people that they are not needed, they are not needing this treatment. So we need a very safe profile, and this requires bigger studies. At the same time, maybe the thing that we are talking and could be possible is to target certain groups that we consider that they have a bigger risk of the disease. We can target, for instance, people that they have a positive serology, or specific people, specific groups of population with really high risk of the disease, like certain activities. So we are envisaging the possibility of targeting these groups, but for that we need to be sure that we have a very safe drug and we are not causing more harm than benefit. No, it's very important to assess this. So I think that there are now research work in progress to assess the safety profile in deep of the new drug acosiborol, to see in a certain moment we can use like this. Important to say also, if we are changing this approach, it means the other things will change, and we need to start to think how we adapt to these changes. Imagine we are treating that is not for tomorrow, but maybe in medium-term future, we are treating these people just with a rapid test positive. We need to know, for instance, for how long we need to treat these populations, when we have to stop this extended treatment. I prefer to talk about extended treatment more than mass treatment. Or for instance, what is the real situation? What are the real cases? All these people we are treating, they are serological positive, but who are the real cases? We need to do some adaptations and to know what is the epidemiological situation, because if we ignore what we are doing, it can be very risky. So we need to adapt other tools to this possible situation.
Okay, thank you, Dr. José Ramón. Professor Simon, could you complete?
Oh, I agree with what José is saying, that this is a critical thing. I think also what he's saying was about what people have picked up on the serological tests, and both his review and that review by Philippe Boucher in Lancet, point out the number of false positives that the current serological tests pick up. And I think this is one area where it wouldn't, if it was a safe drug, it wouldn't matter, because then you could just treat those false positives with a degree of knowledge that it's safe and you're okay. But a lot of studies have to be done to ensure that that is a safe drug, not just adults, but children as well. And that's an important thing. And with that safety, will have to come, and I'm sure DNDi and your colleagues, Florent, will be following this up. There has to become, the clear understanding of the difference in pharmacokinetics and toxicokinetic profiles between children and adults. That has to come as well. But I'm slightly more positive. I think the experience with malaria in the same regions where single dose control drugs, chemoprophylactics, have been used, I think it has caused obviously a major change in the way malaria control programs are planned. And if this drug does come through from DNDi, I think one of the big research things between now and 2030 is to ensure that they can be rolled out for both treatment and control. And the right sort of research program has to be in place to ensure that.
Thank you, Professor Simon. Another question from Venezuela is coming from Venezuela. They say that this question is addressed to José Ramón. There are less cases of HAT. I have the following question. Are there asymptomatic cases in HAT? How big are the numbers? Will this impact the situation in the future? How this situation would impact in the future the identification of infected patients, and how can we maintain the expertise of the physician and other people related to the health system? José Ramón.
Yes, sorry, I was muted. Thank you for the question. So it's true, we know there are asymptomatic people infected but not showing symptoms. That is well described. How many? We don't know exactly how many. Nobody knows, but we think it's not a huge number. We know that it depends a lot, but there are some people that they can develop the disease after many years of being infected, and there are others even that they can be cured spontaneously. But majority of the people that they are infected, they develop the disease in the coming 1, 2, 3 years. We know that. There are more and more research that is going to check if there are about the possibility of trypanosomes hidden in either in the skin, in other tissues, and I think it's important. But we don't think it will be a huge number. At the same time, well, they will not play a big role to maintain the high levels of endemicity, but these individuals, they can play a role to maintain very low levels of endemicity, or even to the re-emergence of the disease after we are not seeing cases for some time. They can play a role in this sense. Then we are thinking, and I think it's a room for research strategies for addressing this possibility. One of them is to treat serological positive. In this sense, you will be treating people without symptoms but infected. This is a possibility, and new safe drugs, as we have been talking before, they can play a role in this issue. And also, well, I think is to identify the burden of this group of people, and the possibilities to identify these carriers through serology, through other methods.
Thank you, Dr. José Ramón. Another question, likely sustainability will be guaranteed through making full use of the new drugs, continue working on safety profiles, so to allow having a label which allows treatment also in case of suspicion. Then a small stock of experts should be concentrated at the Ministry of Health, maybe in conjunction with a reference lab, which arrange and drives constant and continued education of the healthcare staff in all affected regions. Without full integration, financing of activity will likely become very challenging. José Ramón.
Well, I think I understand the question in the sense that, yes, we'll have less and less expertise, and we are working in this sense. I think with the new tools, we are looking for the way to use these tools, but people that they are not experts in HAT. There are people, they can do a rapid test for HAT, for malaria, for other diseases without problem. So it's to integrate some activities in the peripheral health level with people that they are not experts in HAT, they are working in primary health care. But yes, we need to keep some expertise, and we'll need to use very well this expertise, people knowing to do a proper microscopy, or as it was said, referral samples. We have to play with these elements. Maybe in the future, if we need to confirm a case, somebody that has the expertise and has a very high level of expertise can go to the patients, but when the previous screening has been done by the primary health care workers. So we are playing with this integration of some activities, but keeping some capacity of expertise, yes, mainly as it was said at central level. And more reference labs can play a role in this process. It's, as I was saying, we have to adapt these strategies to this new epidemiological situation, and with the tools we have available.
Thank you, Dr. José Ramón. Professor Simon, you have anything to add?
Thank you, Florent. Yeah, I think one part of the question sort of referred to cost as well, I think, of the whole process. Obviously, if you're making, like with albendazole for filariasis and other helminthic diseases, if you are making sort of a batch of drugs and you're making 10 million tablets at a time, the cost is a lot cheaper than if you're only having to try and potentially think about treating 10,000 people over a 3-year period. So the cost of a drug, maintaining a drug supply for a rare disease is very high. And I've had a lot of experience recently working in visceral leishmaniasis on the elimination programs, with the elimination programs, and how this can impact. Obviously, donation helps. But I think this area of cost and the cost-benefit analysis is very important. It is an area where more research is needed. There was a paper published last year in PNAS by, I think it's the first author was Antillon, I don't know this person, about the cost-benefit analysis of actually treatment and dailies in relation to HAT elimination. I think now as we start to look at the most probably sophisticated surveillance tools, improved diagnostics, and these sort of single-dose drugs, we should update and modify that sort of cost-benefit modeling, because cost-benefit analysis is one of the key issues. I always follow the elimination document, the mini Bible for elimination, the article by Walter Dowdall in 1998 from a conference, and he sets out the clear criteria for elimination, and I still think those are relevant. And two of them that are often ignored. One is social and political criteria and social and political commitment, but the other is cost-benefit ratios. And actually, I think with the new tools coming through, with the reduced numbers, this sort of analysis, modeling analysis, and more research should be done all the time, updating as new knowledge comes along. I think it's an important area. Thank you.
Thank you, José. But an additional question is, when talking about extended treatment, what will be the price of an acoziborol treatment? Maybe, I don't know if José Ramón can respond.
I think nobody can respond. Maybe Simon can say something about. But as you were saying, it's important. We have the experience with the eflornithine, with the other medicines we are using, that we are reducing. We are using much less now, but the price is not reducing in the same scale, no? Because we have to produce small amounts that is more expensive to produce small amounts, and even you reach a moment that is not possible to produce smaller amounts, and you produce medicines that they are not used. So it's not directly linked, but I think Simon has more expertise than
José, thank you. But yeah, the experience from the world of visceral leishmaniasis and the progress in India and Bangladesh and Nepal towards elimination has been really supported by the company Gilead, who's donated Ambisome, this liposomal formulation, a single course of treatment for visceral leishmaniasis. That's based upon donation. I think the people in leishmaniasis worry immensely when that donation ceases, because it's an expensive, very expensive formulation if you want to use it in a private clinic. And so that important relationship between WHO, José and his colleagues, and the pharmaceutical industry is going to be critical for the future. So I would say that the cost has to be zero to the patient.
For the patient,
or,
yes.
Or one CFA, just a marker price.
Yeah, I wanted to remark here, if you allow me, Florent, one thing. It's very important. We are very, very privileged in sleeping sickness, and we're very peculiar situation. Now, for more than 20 years, we have had available all the medicines we have needed. This is not happening in many other diseases. We are in a very privileged situations that we have to keep. We have very committed partners from the pharma industry that they have been donating for more than 20 years these drugs. And we have allowed to treat all the cases because drugs were available at zero cost. This is very important. We are very privileged. We have to keep this situation, and we have to encourage our pharma partners to keep on working in this way.
Okay, thank you, Dr. José Ramón. Thank you, Professor Simon. I get back to Macana. Dr. Macana, tu as retenu les questions que je vous ai posées sur l'expérience de l'Angola en termes de rationalisation du dépistage actif et combiné au dépistage passif et également avec le support de la lutte contre le vecteur. Et si vous rencontrez des difficultés sur le terrain qui peuvent faire l'objet des gaps qui pourront faire l'objet, qui peuvent être approfondis et investigués pour des solutions futures après des études qui pourront être menées. Oui, Macana? Le son n'est pas toujours bon. Hein? Le son n'est pas toujours bon. Il faut arranger ton micro là.
Ou non, peut-être si tu enlèves le vidéo, peut-être, essaye. Essaye de couper la vidéo, Paulo.
Oui, allez-y. Non, il y a un problème de micro. On ne vous suit pas très bien. Essaye d'arranger un peu ton micro là. Oui, en attendant, il y a une dernière question. Is the surveillance pointing to any increased cases finding in the countries during the pandemic over the past 2 years? José Ramón?
Sorry, Florent, I was looking at the questions. I didn't hear.
The question is in the chat.
Yes,
the last.
Tell me which one. You can repeat maybe.
Is surveillance pointing to any increased case finding in the countries during the pandemic over the past 2 years?
Well, we will see this in some time. Yeah, we are starting to see. In fact, yeah, we know mainly in 2020, there were some activities that they were delayed, they were canceled, some active screening was not possible to be done, some staff was working in COVID and not in HIT, and well, we have an impact that it was not negligible. Nevertheless, I think we saw at the same time that the national control programs, the control of sleeping sickness, despite all the weakness it has, it was resilience to keep on doing activities. Last year, we have 600 and something cases, and it means, sorry, in 2020, and it was a decrease bigger than expected. For this year, well, we have some initial data, we don't have the final data, but it's probable that we have a small point going up. The question is, yes, last year were less activities linked to this problem, but I think we can easily, or taking some time, to readjust the situation. We have a very good experience with Ebola in some countries like Guinea. Guinea was a very good experience. Suddenly we have a decrease in case detection, but immediately as activities were retaken, these cases were detected and it was an increase for in few time to come back to the expected situation.
Okay, I don't know if Professor Simon or Dr. José Ramón, if you have something missing during the discussion, you can add.
Simon, maybe, or I start as you want, or Paulo. well, I start maybe.
Macana, tu peux parler?
Oui, maintenant, je pense.
Ok, ça va. Oui, allez-y.
Oui, bon, merci. C'est vraiment difficile. J'ai tenté maintenant avec mon téléphone. Je puis entrer avec le téléphone.
Ok, allez-y.
C'est vraiment très intéressant. Nous pouvons partager notre petite expérience que nous avons commencé en 2017 sur l'élimination. Nous avons commencé avec l'intégration des activités de la trypanosomiase dans les unités sanitaires parce que dans les temps, l'ICCT travaillait seulement avec les centres de diagnostic et traitement. Donc, il y avait des techniciens formés seulement pour la trypanosomiase. Ils ne pouvaient faire aucun autre travail, rien que la trypanosomiase. Mais avec la baisse de la prévalence, comme il a été dit, nous avons constaté qu'il y avait plus de malades et puis les techniciens étaient presque, ils commençaient à abandonner les services pour faire les champs et autres choses parce qu'il n'y avait plus de campagne. Maintenant, avec l'arrivée des tests rapides, nous avons commencé l'intégration. Comme c'était une outil très facile à utiliser, ce n'est pas comme CAT qui est légèrement compliqué. Avec les tests rapides, les gens sont déjà habitués. Nous avons commencé avec l'intégration, avec les tests rapides. Et qu'est-ce que nous avons fait? Nous sommes dit, comme il n'y a pas l'argent avec la crise du pétrole, nous avons dit nous allons réduire les équipes. Premièrement, réduire les équipes, laisser les équipes nationales aller former les gens sur le terrain et vous envoyer 1 ou 2 techniciens expérimentés qui vont encadrer ces techniciens locaux des unités sanitaires qui ont été formés à l'aide des anciens techniciens de CDT. Et ces anciens techniciens de CDT sont partis dans les hôpitaux, dans les centres de santé pour travailler dans l'horizontalité. Après cela, qu'est-ce que nous avons dit? Comme il n'y a pas beaucoup d'argent, les campagnes ne se feront pas ou bien les prospections ne se feront pas comme dans les temps. Nous allons cibler où nous avons les tests rapides positifs, c'est là où nous allons envoyer les équipes. Donc, c'est une prospection réactive. Ce n'est pas une prospection active que vous allez comme ça, non. Là où nous avons des signaux, nous avons des TDR positifs, nous envoyons les équipes parce que les centres de diagnostic, les centres de parasitologie, on n'en a pas beaucoup. Donc, on n'en a pas beaucoup. On a beaucoup de centres avec les TDR, mais les centres avec la parasitologie, il y a peu de centres parce que les centres doivent être bien formés. Et qu'est-ce que nous avons constaté? En 2018, on a rencontré beaucoup de malades. C'était la première réaction qui nous a interpellés. On a eu beaucoup de malades en 2018. Mais en 2019, il y avait une baisse des activités parce qu'il y avait l'instabilité des techniciens, l'instabilité des cadres. On a changé la direction, la nouvelle direction doit s'imprégner, doit apprendre, doit connaître. Et l'année 2019, on a fait un fiasco. Un fiasco parce que les orientations n'étaient plus comme à l'année 2018. Donc, si vous voyez nos données, vous allez constater qu'il y a une chute du travail en 2019. Et en 2020, avec la COVID, en 2020, avec COVID, qu'est-ce qui est arrivé? Et avec l'état d'urgence, on ne pouvait plus aller dans le terrain. Il y a accumulation de tests rapides positifs dans les centres de santé. C'est ça l'avantage de l'intégration. Malgré la COVID, les unités sanitaires ont continué à travailler et vous comprendrez en passif ou dans les unités sanitaires, le nombre de malades n'a pas vraiment bougé. Mais en prospection active, il y a une baisse des cas. Maintenant, l'année passée, 2021, on a renoué avec les activités, on a suivi les mêmes stratégies, on va envoyer les équipes là où il y a les TDR positifs. Donc, la stratégie combinée pour nous, nous avons trouvé que c'est vraiment bénéfique. Et il y a la réduction des dépenses d'argent parce que nous, on n'a vraiment pas de partenaires, seulement Fund qui nous appuie et l'OMS qui nous appuie avec 4. Fund nous donne les TDR, mais En vertu de la stratégie combinée, FAND aussi nous appuie des fois avec les TDR. Maintenant, l'autre manche des problèmes, c'est la lutte antivectorielle. Nous avons fait une expérience à Banga, où nous avons visité un village où il y avait beaucoup de porcs, et dans ces villages où nous avons noté beaucoup de malades. Et cette année 2022, nous sommes dit on doit travailler avec l'équipe de vétérinaires pour voir comment il faut travailler ensemble, examiner les animaux domestiques comme les panélistes d'hier, on en a fait. Donc, dans notre stratégie, on fait l'intégration, c'est-à-dire les dépistages passifs, non pas seulement dans les CDT, dans les unités sanitaires, et puis nous faisons la prospection réactive. Et dans cette prospection réactive où nous rencontrons beaucoup de cas. Maintenant, notre préoccupation, c'est l'investigation, c'est quoi? C'est les recherches. Nous avons beaucoup de suspects sérologiques. Nous en avons beaucoup. Comme Dr Simon avait dit là-bas, la sécurité des médicaments. Comment pouvons faire? Pouvons-nous traiter les suspects sérologiques? Maintenant, cette année 2022, nous voulons suivre tous ces suspects sérologiques. Après combien de temps ils peuvent positiver ou ils ne vont pas positiver. Donc, est-ce que s'ils vont positiver, ils vont transmettre les malades et comment nous pouvons arriver à l'élimination? Parce que nous avons beaucoup de suspects. Prenons par exemple l'année 2021. En Angola, on a 765 suspects sérologiques qui n'ont pas été confirmés. S'il y a quelques malades là-bas, c'est ça les grandes préoccupations. Nous ne pouvons pas soigner les suspects sérologiques que nous croyons, nous faisons les 4 ou bien les TDR positifs, les TDR sont positifs, malgré la parasitologie est négative. Ne pouvons-nous pas soigner par rapport à la clinique de ces suspects? La clinique ne peut pas intervenir avec l'expérience des médecins là dans les zones endémiques où nous voyons que, exemple, Banga, nous avons beaucoup de malades, Bolongo, Kilkulungu, ce sont des zones qu'on avait abandonnées. Mais il y avait seulement un malade que nous avons qui était diagnostiqué en passif à Kilkulungu. Et quand nous avons envoyé l'équipe, c'est 27 malades. C'est 24 malades. C'est vraiment, ça nous a beaucoup interpellés et beaucoup de suspects sérologiques. Et c'est ça vraiment les grands projets que nous avons. Si on pouvait avoir des partenaires pour chercher nous pouvons traiter les suspects sérologiques, on peut discuter avec notre ministère de Santé. Si les comités d'éthique peuvent permettre de faire cette expérience, je pense que ça peut nous aider vraiment pour arriver à l'élimination. Parce que les suspects, vraiment, c'est un grand réservatoire des malades.
Merci, Dr. Macana. Je voulais, I want to get back to José Ramón and Professor Simon. You can give your comment about serological suspects in endemic countries, and also about how we can balance active screening with passive screening in endemic countries. Yeah. José Ramón.
Thank you. Thank you, Florent. Thank you, Paul. Nice to listen to you. Yeah, well, we have this situation that is not new of the serological suspects. It's the history that we have been facing in different ways in the different time. That's the important thing. We have to adapt the situation, to adapt the decision to the situation we are getting. In an areas where the endemicity, the prevalence is high, we can consider to treat these patients because, well, as it was talked before, there is an important benefit. It's also linked to the safety of the drugs we are using to treat these patients. It is not nothing new. It has happened in the past, but it's important to do this. And the important thing is to know better in the future, to invest, to know better what a serological positive means. We know that in areas of low prevalence, majority of seropositives are not real cases, are cross reactions. That is also well described, but it's different in the different situations. So it's important. The thing I want to remark again is we need to adapt to the different situations. Different epidemiological situations can carry different case management decisions.
Professor Simon?
I think in this case, José has the expertise, and I have to agree with him. I haven't got the expertise in surveillance of this disease in Africa, and I refer to him as the expert.
Okay, another question. HAT can be seen as a sentinel to socio-environmental disruption and ever-changing public health priorities. Considering the forecast change, how do you plan to sustain the success at a global level in face of lingering cases, lower interest in technical staff or overall community, national disinterest or donor fatigue in the year after the next goal posts? José Ramón?
Yes, well, I will try to answer a part of this question. Well, as we were saying, we are now in a situation that we can consider in majority of the endemic areas that sleeping sickness is not a public health problem. So there are other priorities that they are real public health problems, but is for that we are facing now a different goal. We are facing elimination. So we have to use this to say we are not dealing with a public health problem, I don't know, malaria, HIV, or the many other diseases. It's a real public health problem for this population, but we can get a lot of benefits with certain support, that is to eliminate a disease. I want to come back to the first talk of Professor Anderson the first day. Only 2 diseases have been eliminated in the past. There is a third one that has been targeted for elimination some years ago, is Guinea worm, even a disease that has not vaccine, but even more, it has not treatment. So this disease was targeted for elimination in 2000, still 2020, and they are fighting with the last cases that they are quite complicated. I want to underline this to say, well, we have a big challenge. If we go for elimination, we have a big challenge. It's not easy. The last cases are usually difficult. But I think that it will be a huge benefit if we reach to eliminate this disease, to be sure that in the future we will not have again this disease that in the past has been ravaging many populations. We have all the community, researchers, funders, donors, all the stakeholders. If we have this idea, I think we can keep this to avoid this donors fatigue, to keep countries interested in this disease. Is for that well, to this target of elimination is important as a goal, because well, it helps to keep people involved.
Professor Simon, anything to add?
Yeah, I'd just like to add that I think to sustain this, to stop the fatigue, etc. I've again come back to an example I've been involved with observing in Kala Azar, visceral leishmaniasis. I've been very impressed by how Bangladesh has come very close to the elimination of Kala Azar, certainly as a public health problem, and probably going for zero transmission. And I've been impressed by, from the top, there's been both leadership and there have been champions. And there's a difference the nature of these two different groups, but Bangladesh has both leaders who make clear decisions and champions who make sure that they do make the decisions and don't sort of go off target. And this, from this leadership, it's percolated down the system. So there is a pride, not just at the top, but there's a pride in the centers for research, in the Ministry of Health, in the disease control program. There's a pride in going for this elimination. And there's a pride that when I have had a chance to visit villages, to ask them about the disease, what they understand about the disease of the community, they do know it's transmitted by a small fly. There is a pride there, even in the villages, that they are understanding and going to do something about the disease. So it really is part of the national system of leaders, champions, and the pride in going for that elimination. And that has really, I think, in Bangladesh, even though the numbers are very low, that there is no fatigue there, that they're going for it. They're going for it, and even trying to get zero transmission, not just elimination as a public health problem. So I have seen it. It is possible, and obviously there are some African countries I wish well in adopting the same sort of approach. I think it would be excellent.
Thank you. I think that we have two last questions regarding the new drug to be used. If I understand correctly, the treatment is for 10 days. Regarding the viability problem that we are discussing right now, how big will be the possibility of drug resistance emergency since it is a treatment with one drug only? Professor Simon.
Okay, well, thank you. And thank you to very old friend Alicia for asking this question from Venezuela. Alicia is an international expert on drug resistance in leishmaniasis, and so she asks the right question. The 10-day treatment from Sanofi is fexinidazole, and that has been approved and is being rolled out and is being used in the treatment of West African Trypanosoma gambiense, and José will be the person who can tell more about that. The other drug I was referring to is completing this phase 3 trial. There's been an 18-month follow-up, and I think one of the key things is when this is the potential single course treatment, acoziborole, which could be used at community level. Now, fexinidazole is, as Professor Pontrasico will know, it's a drug which there is a known target. It's a drug which is actually activated. It's a prodrug activated by nitroreductases. And so there is a potential for drug resistance to arise if the drug is abused. But if the drug is kept, I think, focused and through community action, both compliance at the community level and adherence at the patient's level are kept high, and then the health system ensures there is a sort of concentration of use of that drug around the foci of infection villages, I think there is a chance of actually ensuring that there is no resistance developing to fexinidazole. But it's with drug resistance, as Alicia will know, it's always the famous last words. There will never be drug resistance, but of course, drugs and the microorganisms are always cleverer. They always find a way somehow. It's a matter of ensuring proper use and proper control through the health systems and the communities. And I'm sure that is a message that came across yesterday in the community discussion, and I hope will be taken on. And I'm sure WHO and José will actually be, and you as well, Florent and Paulo, will be part of that system.
Okay, thank you very much. I can read a comment coming from Gerardo Priotto. As pointed out, acoziborole, the safety is a cue for widening treatment to individuals suspected of infection, but not confirmed. Is that gap going to be filled by the studies currently planned? Will we be able to treat most of the population, including children, reproductive age women? Should more study be planned? I don't know if Professor Simon have a comment.
So yeah, I have a comment, which is Olaf is probably online somewhere. I see him here. I think Olaf is in the best position to comment on that, as he's in charge of these things at DNDi.
Okay, Olaf, could you comment?
This is a betrayal, because I was not supposed to talk in this meeting. Yes, so I think that with the current trial we are doing with not only with cases but with seropositive individuals, we will be in a very good position to be confident about safety, because we will include 1,300 or 1,200 people, I think, in total, of which 900 will be treated with acosiborole, but we are blinded, so we will know at the end. But this, together with the previous pivotal clinical trial, will give us a good number of people. That's one. The other one about children, that's more difficult because you know, we are planning to start very soon a clinical trial in children, but the problem is that we will certainly find few. And we hope that with this data, we will be able to extrapolate the much bigger information we have with adults. And regarding reproductive age women, this is the same problem we face with every trypanosomiasis drug. We will never join sufficient pregnant women to be sure that we can use it freely, but this is something that remains open. Thank you. Florent, we don't hear you.
You're muted.
Okay. Another question, another comment. How important are animal reservoir elimination so much more difficult if they are significant? I don't know if Professor Philippe Boucher can tell something about it. I don't know if he is online. Maybe José Ramón, could you respond to this question?
Yes, as you say, well, Professor Boucher has much more expertise in that, but it's true. Well, the thing I can say from the public health point of view is that animal reservoir, we know that it's possible to find parasites in animals. We don't know the importance of that, but it's true that parasites can be there. It can play a role. Probably not important in maintaining transmission, but it's important in appearing cases, in reemergence of the disease. And it's for that we are not going for eradication of the disease. We are just trying to eliminate the disease because it's possible. Maybe Philippe can tell more in detail. It is possible always that is remaining a cycle of the parasites in animals. So it can be always be a reemergence of the disease. But I see that Philippe is now there. So.
Yeah, okay, thank you. Do you hear me?
Yeah.
Okay,
yeah.
fine. Well, it's true that we do not know the role or the significance in transmission of an animal reservoir. We know that Gambiense, for example, can circulate in animals, but we do not know how important that is for the transmission to human. But when we talk about reservoirs, I would focus more on the human reservoir, since they may be much more important even when the number is low. As long as tsetse flies are around, these undetected cases, for any reason undetected, that could be that the serological or molecular tests are not sufficiently sensitive, or that these persons are just not seen by screening activities, etc. So I think this human reservoir is much more important and much more dangerous for an emergence, re-emergence of the disease. So I would focus on studying more the human reservoir than the animal reservoir.
Thank you. But it is the last. I don't know among participants who are following us. Do you have anything to share with panelists and all the participants? I don't know if someone has something to add or to share with us. If no, we can give some remark about this session. I think that all the priorities and research gap will be gathered by the organizing committee. Maybe they can share with all the participants who attend this meeting. José Ramón. I see the hand. José Ramón?
Yes, I don't know if you are going to end or we can say something still.
Okay, you can go.
No, I want to do a couple of reflections from my side. Maybe they are useful for the others. I have to say that I'm not a researcher, I'm a public health officer. But I think it's important. We have limited resources for HAT. We are not a public health problem. We cannot get all the resources we would like, but we have some resources. And the question is that, where do we put? We put in research, we put in control. I think this is very important to do a balance. We can not put everything in research, we cannot put everything in control. It's an important balance, so we need to prioritize. And it's for that, this is very important, the coordination between the people working in the control and people working in the research. This, I think, platform, the HAT platform is working in this sense. WHO is coordinating all the stakeholders. We are trying always to put together researchers and people working in the field, in the control of the disease. And it's, I think we in a certain way, we are succeeding to do that. We have to keep on doing that. And that the research is answering the needs of the control. So we have to work always in this sense, this balance. We can't put all the resources in one side or in another side. So I think in this sense, this meeting is interesting. And I want to bring another point related to that. Sometimes we are developing tools, but it's important later that we guarantee that these tools will be accessible. Sometimes we can develop tools, new treatments or new diagnostic tools or new vector control tools or whatever, but there are later, the populations suffering the disease can later access these tools is not always sure. For the treatment, we have had the situation that the pharma companies have been accompanying the process and they are offering later this treatment. It's not the case sometimes for the diagnostics. We have developed interesting tools, or there were some tools in the pipeline that later on they have not gone later because it was not a commercial partner supporting this, the production, the affordability later of these things. So this is also an important point to be taken when we are talking about research and needs to ensure later access to these tools developed. And just one final point from my side, we focus a lot in Gambiense, is the big part of the problem of trypanosomiasis, but we don't have to forget Rhodesiense trypanosomiasis. More linked to animals, more approachable from one health side, but it's important to say, currently treated cases in second stage of sleeping sickness, Rhodesiense, are treated with melarsoprol, that we have been always complaining about its toxicity. we don't have serological tools for rhodesiensis. So we have very few cases, but it's very neglected disease. And we have also to always keep in mind that there are some needs in rhodesiensis. So thank you very much, Florent, and London NTD Research Center for this meeting.
Okay, very good point you mentioned. Maybe Professor Simon, your feeling during this roundtable?
I think I commented actually earlier that I learned more because of being a new area for me, an area I'm very much involved in development of tools for 40 years. So yesterday's discussion about the role of communities, about communication within the communities, about learning at that level, I was very pleased to learn more, and I think I realized this was an important area because my work, similar work in leishmaniasis, but to hear it discussed in such depth yesterday, I think really did bring the point home. So I was very pleased to hear yesterday's discussion.
Dr. Macana, vos impressions par rapport à cette table ronde? Est-ce que les attentes ont... Oui, Dr. Macana?
Merci beaucoup, ça a été vraiment intéressant. Je pense que c'est un grand objectif que nous avons d'éliminer la maladie du sommeil. Malgré le chemin est encore, je vais dire long, c'est comme Dr. Franco vient de dire, les contrôles et l'investigation. Je pense qu'on doit travailler ensemble avec les partenaires. C'est comme notre expérience en Angola. Il y a quelques études que nous avons faites seulement avec l'appui des partenaires. Donc, maintenant, dans ces derniers virages, j'espère que tous les efforts doivent être mis ensemble pour voir comment nous pouvons arriver à l'élimination. Donc, comme je viens de le dire, notre petite expérience en Angola, nous voulons continuer à l'exercer et puis nous cherchons ces partenaires qui peuvent nous booster pour arriver à l'élimination. Moi, je dis toujours Angola est capable d'éliminer la maladie parce que nous avons la volonté, le gouvernement a la volonté et si les partenaires peuvent se joindre avec nous pour la recherche et pour les combats et pour les prospections, je pense qu'on y arrivera à retrouver les dernières maladies et arrêter la transmission de la maladie. Merci.
Merci. I think that we are at the end of this roundtable. I would like to thank all the panelists. And then I get back to Roy for closing remark. Roy?
Fine, thank you. I have two tasks I'd like. First of all is to thank all the panelists, both on the previous 2 days and today. I've learned an enormous amount. I'm not a specialist in the protozoan neglected tropical diseases, but I remember as a young student always being hugely impressed at looking at trypanosomes down the microscope at slides and wondering at the sophistication and complexity of the beast. Your problems, in some senses, listening to this from the perspective of other infectious diseases, your problems are in part one of success. You've been hugely successful in reducing the prevalence of infection and identifying hotspots, setting up effective surveillance systems and so on. And inevitably, as the disease becomes not a public health problem, people lose interest. And that's a classic problem, which we're going to see with a number of other NTDs in the coming years, the next decade. Now, how we maintain interest, I think, is that this is still a very serious infection for individuals. And one appreciates hugely Sanofi's maintained interest in developing therapies for this, and indeed for helping us support this meeting today. So long may Big Pharma continue, as Simon mentioned, the drug donation side. And clearly there's been such progress in the development of the new drug, and let's hope the clinical trial on the safety side is very positive. There are enormous opportunities then to really get towards the elimination, but it will be a matter of, as it were, keeping our paymasters' interests in this field very much alive. The other two problems that struck me quite forcibly was that the science technology side of diagnostics, number one, and number two, vaccines, has changed out of all recognition in the past 5 to 10 years. And it would seem that as a disease becomes rare, ironically, diagnosis becomes more and more important. Having sensitive and accurate diagnostic tools, one can see it in a vast number of different areas. Of course, cheapness of the diagnostic is an issue. And these more sophisticated methods, which are more sensitive, of course, tend at the moment to be more expensive. But it struck me that you weren't totally comfortable with the sensitivity and specificity of the tests that are available at the moment. And that would seem, as the disease gets rare, a very, very important priority, which is probably soluble given modern technologies. The second issue is vaccines. The template for vaccine development over the past 5 years, 10 years have been staggering improvements. And as some of you may have seen, Moderna thinks it's got something very helpful for HIV as of this week, which is going into human trials at the moment. And then we're very well aware of the mRNA vaccine development within 6 months of something effective against COVID. Now, if you've got an animal reservoir, then actually that's quite helpful in some ways, because with vaccine development, the animal can often provide a very useful target at the beginning for developing a vaccine. It's much quicker to do it in the veterinary context. So those two things struck me very forcibly. The one that probably is most worrying was the one mentioned by José, which is a common in a variety of vector-borne infections, and that is the loss of expertise, whether it's on the entomological side, whether it's on the trained technical and public health workers to detect disease in natural endemic settings. And that's a huge issue. And I think all of those still working in tropical medicine really have a duty to make sure that there are young people coming through who sustain an interest in these infections. Lastly, so those are just some personal thoughts. I've enjoyed this immensely. I very much agree with Simon's comments. The social and behavioral side yesterday was extremely interesting. The storytelling aspect. How do you, with a disease as complicated as this one, how do you get communities sensitized to helping in the identification, the surveillance and monitoring as the infection becomes rare? And let's hope some developments can occur there with very good sort of storytelling modules, which can be, as it were, taught to the healthcare workers in those settings in the villages. Now, lastly, my thanks, I've mentioned Sanofi, and thank them very much for their interest and support of this. And long may you remain interested in the protozoan infections and the treatment of them. But could I also very much thank our collaborators, first the HAT platform, who were exceedingly constructive and helpful in choosing speakers and so on. And then also the Royal Society of Tropical Medicine and Hygiene in the UK. Thank you very much for all your technical support for this. And then I'd like to thank Catherine Forbes from the London Centre, the manager, who has put a great deal of effort. So Catherine, thank you very much indeed for all your hard work here. But again, just to end, success has its penalties, and let's all of us have a duty to try and sustain interest. And this looks like a disease that essentially you could eradicate, given the progress you've made over the past 2 decades. But thank you all very much indeed for your interest. It's been a great learning experience. So I say goodbye. Thank you all very much indeed.